You can start.
Hello, everyone. My name is Or Kahlon, and with the Hebrew University Medical Center and the Faculty of Medicine in the Hebrew University of Jerusalem. And I'm going to tell you today about our novel small molecule, which we designed as a therapeutic agent for APBD, adult polyglucosan body disease. Right. Okay. Before I start, I just wanted to give you a general description of our experimental approach. We are actually champions of understanding the mechanism of action and as a condition for the development of pharmacological therapies. So we start by a screening approach in which we just define or discover a clear readout. Okay, once we have a clear readout for identifying our therapeutic agent, we go to thorough exploration of the mechanism of action. Which means we know with which protein it interacts and what is the cellular pathway that it modifies. And only then do we go on to see if it affects the histopathology in the disease. I'm showing here APBD, but again, this is the general approach. And then we go through in vivo studies. You see an example here. And only then we go to clinical trials. So this is our general approach. It doesn't have to follow the timeline, but we do require from ourselves that we go through all these steps. So if we talk about APBD, adult polyglucosan body disease, the pathogenic factor, as many of you may know, is aberrantly structured glycogen. So normally glycogen is a highly branched molecule, as you see here, which generates this global huge molecule of more than 50,000 glucose residues, and which makes it soluble in the cytoplasm. However, when you have less branching events or less frequent branching events, which is what is caused by the branching enzyme deficiency, you get a more elongated structure like that, which looks more like starch than the glycogen. And this actually is considered the pathogenic factor. It accumulates in cells, but especially in neurons or in long axons, long and narrow axons, it can precipitate and clog them. And this causes the APBD symptoms. So basically, there are two approaches actually to tackle the accumulation of the polyglucosans. The first approach is changing this activity ratio. So whenever you decrease the elongation by glycogen synthase or increase or activate the branching of the branching enzyme, the equilibrium or the balance between the polyglucosan to glycogen tips towards the glycogen at the expense of polyglucosan, okay? But I wanted to stress that this actually refers to the formation of the polyglucosan, okay? So you reduce the polyglucosan formation de novo, okay, if the enzyme activity ratios changes. So there are approaches, there are also approaches in other diseases where you inhibit glycogen synthase or inhibit the elongation, or more rare approaches that we also took, you activate the aberrant branching enzyme. Another approach, which we think is more effective, is just to degrade the pathogenic factor, to degrade polyglucosan. Why is there advantage to this approach? Because it actually tackles all the polyglucosans that are there, and not only the new ones. In this approach, you only affect the formation of the polyglucosans. The pre-existing polyglucosans which generate damage are not affected, and actually you have to wait until they degrade spontaneously, so that you don't have the pathology. So in this aspect, this approach is preferred. However, as I said, we didn't know a priori in advance what would be the result. Our result is just to look, our approach, sorry, is just to look for what agent will decrease the polyglucosans, and then only then we decipher the mechanism of action. So here is our screening approach. What we did, we started with developing a screening assay in which we took skin cells from patients, ex vivo cells from patients, and we just used a very known assay called the periodic acid sheaf staining to look for polyglucosans. So we degraded them with diastase, and therefore we know that we're looking at polyglucosans and not at glycogen, because the polyglucosans are resistant to diastase digestion. And here you can see what we have in the healthy control and in the patient cells. So obviously, there is accumulation of polyglucosans in all the tissues, and we show here skin cells. So once we have this readout, the next step was to just screen for various molecules. We actually screened for more than 10,000 different, very diverse small molecules, and we looked for molecules that turn this into that. So actually, that reduced the polyglucosans. We didn't know at this stage by what mechanism, but we just selected the molecules which reduce the polyglucosans. Okay, so again, from this condition to this condition, and GHF201 was one of them. How did we select GHF1 from all the positive hits we discovered? So the first step, we did some toxicity assays, and so on. But the first step was actually in silico. Okay, so we just used bioinformatic or algorithmic prediction algorithms to predict which of the drugs that we found is going to be less toxic, less metabolized, longer lived, and so on. So there are some criteria that the chemists or computational chemists can discover in silico. And you can see in this heat map, the more green, the more preferred the molecule. As you can see, on average of three different algorithms, you can see that GHF201 is a preferred hit. So once we established that, we wanted to test that if in real life it is also safe. And I'm just showing you some of the main safety assays. So both in the long term and in the short term, I'm showing you just the short term to make it short after 5 days. When you treat mice with GHF201, with this molecule, you can see there are no lesions, okay, in different tissues as compared to the vehicle treatment. Okay, so number one, there is no lesions, which means that histologically it is safe. And also, number two, no change in the weight, which is a very strong indicator of the animal well-being. So there was no change in the weight. And we also conducted Erwin tests, which is a battery of various behavioral, posture, appearance parameters, and they all showed complete safety of this compound, GHF201. So then, once we established the safety, we started to test this compound. We used the mouse model of APBD in which GBE was knocked in. And actually, well, I mean, I say we, but it's not us. It actually was made by Dr. Rohan Ackman from Columbia University, who kindly shared these mice with us. And these mice actually have the same human mutation. So instead of the murine gene, they have the human gene with the mutation. They call GBE-YSYS. They're homozygous for this mutation. And we injected the GHF1, the drug, twice a week in this concentration, 250 mg per kg, which was limited by the solubility of the compound and the amount of DMSO that we can inject to the animal. And we later on talk about the new mode of delivery that we use now. So the basic results were, number one, it increased considerably the survival of the animals. Okay, as you can see here. Number two, it affected improved the reflexes of the animals. You see here the what we call the extension reflex. When you hang the animal from its tail, it opens its paws. It should open it wide. So this is the test that you saw, and this can be quantified. To what degree the animal can open its paws when hanged from the tail. And as you can see, there is a considerable improvement in the extension reflex, which is interesting because in patients, they also have the MRI shows that we have pyramidal signs, which also actually affect the reflexes in patients. So this is a very important parameter because it is also part of the human pathology. General parameter for locomotion is the open field, and as you can see, affected animals, the mutant animal is almost stationary, doesn't move a lot as compared to the wild type, and you can see a tremendous improvement in its movement. Sometimes they call it compound 8. This is the old name of GHF201. So this is actually GHF1, and as you can see, it significantly improved the locomotion, the movement of the animal. Here you see the gait, and you can see that the animal is actually weak, and it actually has a very small steps as compared to wild type. And you can see again a significant improvement in the step size, which means that the muscles are stronger, and of course the gait is closer to wild type after the treatment. Okay. So these were actually behavioral or motor parameters. However, we also had to demonstrate that this is not by chance, that it is also related to an effect on the polyglucosan in the different tissues. And as you can see here, in the liver, in the cortex, the heart, which is a very important organ because it uses a lot of glycogen relative to other organs, the heart, peripheral nerve, so not only the CNS, also the peripheral nerve, there was a considerable reduction, which we also quantified in the amount of polyglucosans. We didn't see a significant effect in the muscle. Okay, this was also quantified here. Also, there was a reduction not only in polyglucosan, also in total glycogen, which was determined chemically, but we didn't see an effect in the muscle. And this is related to the fact that the GHF201 simply didn't accumulate in the muscle. So we conducted the pharmacokinetic profile, so we injected the compound, and then we followed it over time in different tissues. And as you can see here, while it did get into the brain, into the liver, okay, which is the highest accumulation, and to the blood, it almost didn't get to the muscles. So this is actually in line with lack of effect at the histological level. So it didn't affect the polyglucosans in the muscle, which is can be explained by the fact that the compound simply didn't get to the muscle, which is not surprising because the muscle is poorly perfused organ. And then we actually looked at the in vivo metabolism of the animals. So we used the very advanced metabolic cages, which can measure the movement of the animals, very sensitive methods, the gas exchange, and so on. It's a very sensitive tool. And we saw that animals which were treated increased the level of carbohydrate metabolism, their ambulatory activity, which actually reinforced our results in the open field, and the total energy and expenditure. So I just wanted to go back to basic biochemistry, if you will, and remind ourselves about the concept of respiratory quotient. Okay, so if you look at the fuels that cells use, when they use the more they use carbohydrates, okay, when they burn carbohydrates, they burn them eventually into a pyruvate, which turns into acetyl coenzyme A. Acetyl coenzyme A enters the Krebs cycle or the citric acid cycle, which produces reducing power. This is the most important product of its metabolization, of all metabolization, reducing power, sorry. Why? Because reducing power donates electrons in the respiratory chain, which eventually go to oxygen, and in this process, of course, the mitochondria produces ATP. So the more carbohydrates are metabolized, the more there is tendency to go of the electrons of the reducing power to go to the citric acid cycle, which releases CO2. Okay, you can bypass the citric acid cycle if your fuel is not carbohydrate, it's fatty acid or amino acid. And as you can see here, and this is from a basic textbook of biochemistry, the fatty acid and amino acid oxidation can produce reducing power or NADH directly, bypassing the citric acid cycle. So this means that the ratio of CO2 release to oxygen consumed, okay, oxygen is reduced by this reducing power by this electron, the ratio of CO2 released to O2 consumed is related to how much carbohydrates are metabolized in comparison to other fuels, amino acid and fatty acid, okay? And so when we looked at the respiratory quotient of the animals, we can see that indeed, when we treated them with GHF201, the respiratory quotient increased, which means that as a whole, the animals actually metabolize more carbohydrate at the expense of fats and protein, mainly of fats, because this is the main source of the main other alternative fuel to carbohydrates. So they metabolize more carbohydrates relatively. And as you can see, they also can eat more and metabolize more carbohydrates. So glycogen obviously is the most important candidate, because it means that they can metabolize glycogen, which is the main carbohydrate the cells use for energy, as an energy store, okay, especially in the muscle. And then we also looked at the biochemical panel in the blood, like you do normal blood test, and you can see the most importantly that the hypoglycemia that you have in the APBD mice, which is expected because the glucose goes into glycogen, mobilized into glycogen, but there is less left in the blood as a result. It sinks as glycogen and less remains in the blood. So the animals, as in other glycogen storage disorders, the animal models are hypoglycemic, and treatment corrected this hypoglycemia into almost normal levels. So you can see that the hypoglycemia was reversed by GHF201, and also the triglycerides, which are high usually because there's a high flux of fats into the liver as an alternative source of energy in the affected animals. So the triglycerides were reduced. So it corrected the hypoglycemia, it corrected the hypertriglyceridemia, okay? And it didn't cause liver damage as tested by blood creatine kinase, or muscle damage as tested by creatine kinase, or liver damage as tested by alanine transferase. So it didn't have an adverse effect on the general metabolic parameters, but it improved the parameters which are defective or deficient in the disease model. Now, in patients, you can see here different patients, glycogen levels are very variable, okay? So you can see here, again, fibroblasts from different patients, you know, they're different numbers, and you can see the levels of polyglucosan here, okay, quantified by past staining is very variable. And therefore, since our target, or pathogenic target, is glycogen, we designed the condition we called glycogen burden, which means that the cells that have more glycogen will be more responsive to the therapy, because glycogen is actually the pathogenic factor. And we did it by starving the cells of glucose for 48 hours and then replenishing the medium with glucose. Once you replenish the medium with glucose after starvation, the extent of uptake of glucose is much higher, and therefore there is more glucose which is deposited in glycogen, as you can see here. And when at the cell level, when you look at different organella parameters, like you can see some of them here, some parameters of the mitochondria, the lysosomes, the cells organelles, you can see that they were affected by the disease, okay? This is the this line actually is the control. You can see the deviations from the disease above this line. And you can see the deviations in blue in just in staph cells. When you generate the glycogen burden, you can see there's more glycogen. All these effects actually, all the effects of the drugs were exaggerated. Okay, were enhanced. You can see the red line, and all the differences in the different cell organelles were enhanced by the treatment. Okay, so then we thought, okay, if this is true, that you have actually degradation of glycogen, because we saw that by the in vivo metabolism, we saw that carbohydrates are degraded, okay, and we know that there is less glycogen by the chemical and by the PAS assay. This should mean that actually you should have more ATP coming from glycolysis, okay, than from other pathways, which are the alternative, of course, is OX4's oxidative phosphorylation in the mitochondria. And indeed, again, I show here the different patients. When you treat them, especially when you treat them acutely, so the cell doesn't have the chance to adapt, you can see that more ATP is derived from glycolysis. Okay, and this actually is strongly suggested that the glucose, which serves as the substance for glycolysis, is derived from glycogen. Because we know that glycogen breaks up, we know it from what I showed you, and now we know that more ATP is relatively derived from glycolysis. Okay, so this means that the carbohydrate breakdown, the glycogen breakdown, is also efficient as it can produce ATP. Okay, the fraction which produces ATP is more glycolytic. So this means that not only is the carbohydrate breakdown, it is also energetically efficient and can be translated to glycolytic ATP. It's a very important result. And then we actually looked at the mitochondrial bioenergetics. Before that, I also wanted just to give you a short introduction, so we're on the same page. We talk about modular kinetics. So we have actually 3 types of interference with mitochondrial function with the oxidative phosphorylation. If we look at the basal respiration rate and plot it against the mitochondrial membrane potential, we know that we have uncouplers actually, which cause depolarization, okay, decrease the membrane potential, but at the same time actually increase the basal respiration rate, okay, because the electrons can flow faster as the membrane potential is depolarized. We have electron transport blockers, okay, which reduce the membrane potential and also reduce basal respiration rates because the electrons are simply blocked or inhibited, so they cannot flow so faster. And we have ATPase inhibitor, which actually increases the proton gradient, so the electrons can flow actually slower, and this is why the basal respiration rate is reduced, but it generates a higher potential because the ATPase is blocked, so the protons actually accumulate and the gradient is higher. Okay, and when we do the very basic seahorse bioenergetic experiment, we know there are several parameters that we look at, and those are the basal respiration. Then we add inhibitor of the ATPase or ATP synthase, oligomycin, which causes a drop in the respiration. Okay, and this fraction of the drop, C here, is actually the respiration which is dependent on ATPase. Okay, and this fraction B is the leak. So this is actually the respiration which is not dependent, does not produce ATP. Then when we add the high concentrations of an uncoupler, we get the maximal respiration rate, okay? And there are several derived parameters. The important ones are C over H, so this is the coupling efficiency, how much of the respiration can be used or exploited to produce ATP. And also we have the respiratory control rate, okay, so it's the total respiratory capacity divided by the leak. So the more leak you have, the less respiratory control you have. And the spare respiratory capacity, which is D minus A, which means how much energy the mitochondria has to spare. Okay, what is the maximal capacity that it can provide energy? So, sorry, the slide didn't move. Okay, so these are our bioenergetic results in cells, and as you can see, treatment with GHF201 increased the basal aspiration and increased the coupling efficiency. So this difference, okay, after you add the oligomycin, this relative drop is higher, so the coupling efficiency is higher. More of the electrons flow go, or more of the gradient, actually the proton gradient goes to ATP synthesis. So the coupling efficiency is higher. Less goes to leak, more goes to ATP synthesis, and also the maximal capacity was increased. So actually, if we go back to the modular kinetics, GHF201 worked as a reversed electron transport blocker. So electron transport activator. Okay, because it increased, as you can see here, it increased the membrane potential, okay? And it also increased the basal respiration. You can see here. So it increased both together, which means that it allowed the electrons to move faster, or it was actually an electron transport activator rather than blocker. Okay, next. So what is the mechanism of action at a molecular level? And this means whenever you have a new drug, you want to know its protein target. Okay? It acts on a certain target. And we, in the collaboration with the French company INEPIEM, we looked for this target. And now they designed a very advanced and very sophisticated assay for detecting what is called the heteroassembly, or the quaternary structure of the proteins which react with any given molecular candidate. And this is actually based, of course, I won't go into the detail. It's called the enzymatic protein optimization technology. And this is just based on liquid crystals which form around this molecule that you want to test. Okay, so for instance, here you can see that when you don't add GHF201, there is no formation of a liquid crystal which is made of proteins, okay, which are the targets of this molecule. And you see that when you add it, you do have them, and you do it in the physiological environment. So this happens in true physiology. So it's not a vitamin or any other endogenous small molecule around which these crystals are formed. There is a heteroassembly of proteins which is uniquely interacts with GHF201. So it doesn't mean all of them interact directly with him. It means that this is a heteroassembly. It's a signaling pathway. It's as I said, quaternary structure which is associated with GHF201. One of the proteins can interact directly with him, and the other just gather around him. But this is the heteroassembly, the pathway which is affected by GHF201. And this is shown here, okay, by this string diagram, okay, and you can see all these proteins are found in this heteroassembly, okay, they're found in this liquid crystal, which means they all act together. They are all targets, as I said, direct or indirect, of GHF201. So it interacts with this family, if you will, or heteroassembly of proteins, okay? And then we tested them, what we did next, okay, and we saw that all of them are related to lysosome, also autophagy, so there is actually a functional link between them. And then we tested them just one by one. So which one of these directly interacts with GHF201, okay? And we tested it by an assay called cellular thermal shift assay, which in general means that you heat up the protein as you do with PCR. And if there is binding of the molecule, it protects it from denaturation. So when you look actually at the protein in the soluble fraction, okay, when it denatures, it actually precipitates. It doesn't appear in the soluble fraction. When it's protected from the denaturation or more stabilized, it appears more. So you can see that from all these proteins, only LAMP1, lysosomal associated membrane protein 1, protein on the transmembrane protein on the lysosomal membrane. Only this protein actually directly interacted with GHF21. This was quantified here. Okay, so we said, okay, we know now the target, okay, and we excluded other candidates, but let's explore it more and show now directly, in an accepted method, that it interacts with GHF21, the CLAMP1 interacts with GHF21, and we also know that it is a lysosomal protein. So what we did, we used a very more common technique called SPR, plasmon resonance technique, in which you just bind your candidate on the surface and see if it changes the resonance. I won't go again into the biophysics, but I can tell you that if you have a change, okay, if the plasma resonance signal increases, this means specific binding, okay? And we noticed something interesting. We saw that there was no binding in pH 7. However, in the acidic pH of the lysosome, we saw binding. So the binding got stronger as we decreased the pH from 6 to 5. And then when we got to the lysosomal pH of 4.5, we saw specific binding. This is the plot you're looking for. Okay, you have an increase, and then you have a decrease, or when you have the debinding. So it was specific. However, the affinity was extremely low. Okay, so you can see the KD and the saturation constant was very high. Usually we talk in tens of nanomolars or nanomolars. This is a reasonable affinity, and we asked ourselves, why is the affinity so low? So when you look at this graph, you can see that it's not symmetrical. You can see that the association is very, very slow as compared to the dissociation. Okay, and this means that we suspected that we had a very slow K-on, very slow association. Okay, inside the lysosome, and we thought, why could that be? And then we reasoned, or we hypothesized, that this is because LAMP1 is a heavily glycosylated protein. So there are many sugar residues which are bound to it. And as a result, there is actually a forest of sugars there. And just the diffusion of GHF21 to the amino acid in the protein where it binds is very slow. So we said, okay, what happens if we deglycosylate LAMP1, okay, and allow the diffusion of GHF21 to be much faster? And lo and behold, this is what happens. So after deglycosylation, chemical deglycosylation of LAMP1, the luminal part, we saw a reasonable binding and debinding association, this dissociation, and indeed, now the KD, the affinity constant was reasonable, 52 nanomolars, which made sense. So this structural data actually show you the binding pocket. This is GHF21 here, and this is the binding pocket. The balls here are the sugar moieties, and this is the binding pocket of GHF201 in LAMP1 in the luminal side, and we actually we have this model, this simulation, which actually positions GHF21 between two adjacent LAMP1 molecules, and it actually dilutes LAMP1 on the lysosomal membrane. So effectively, this dilution, okay, is like decreasing the concentration. So like decreasing the membrane concentration of LAMP1, okay? And indeed, we also saw by electron microscopy that lysosomal glycogen was highly reduced by the treatment, which made sense to us because it actually is supposed to reduce the level of lysosomal glycogen, and as a result, total glycogen in the cell. And indeed, what we think the mode of action is actually activation of lysosomal, lysosomal fusion with autophagosome. So this means that actually, that actually when LAMP1 is diluted, more LAMP2, okay, because you don't have LAMP1 and LAMP2, when you dilute LAMP1, the relative concentration of LAMP2 increases, and we know from the literature that LAMP2 can mediate the fusion of lysosomes with autophagosomes. And autophagy is actually a general process by which different inclusions and different waste products of the cells are degraded. Okay, and since we found lysosomes, as I showed before, we found glycogen in lysosomes, we thought that this is the pathway by which lysosome can be degraded, or activation of autophagy, or fusion between autophagosomes and lysosomes. So this actually activates or increases the autophagic flux. So we measure that directly. There is a very elegant way of quantifying autophagic flux. And basically what we tested was actually just the ratio. You have this marker of autophagy called the LC3, and it comes in two flavors. It can be lipidated, LC3-2, and or bound to vesicles, or it can be non-lipidated or cytosolic. So the ratio of LC3-2 to 1, okay, with or without lysosomal inhibitor determines the autophagic flux. Okay, so the idea there is that when you inhibit the lysosome, it cannot fuse well with autophagosomes. So LC3 as a subset of autophagy accumulates. Okay, you can see, for instance, here, without lysosomal inhibitors, pepstatin and lupeptin, you have the ratio of LC3 2 to 1 is low. Okay, when you block the lysosome or the lysosome function, you see that it accumulates, LC3 accumulates and cannot be degraded. Okay, so the idea there is to look at the sensitivity to lysosomal inhibitors, which means that if we have low levels of autophagy, it doesn't matter if you add an lysosomal inhibitor or not, because anyway, the flux is slow. Okay, so the autophagy will not be sensitive to lysosomal inhibitors. On the other hand, if the flux is higher, then it will be sensitive to lysosomal inhibitors, because then when you add the lysosomal inhibitors, it should have an effect, because without it, the autophagy or the autophagosomes fuse well with the lysosome. So the sensitivity to lysosomal inhibitors actually determines the autophagic flux, which is the relevant parameter. Okay, and as you can see, there is with the addition, this is another name for GHF201, 144D11, but it's all the same. You can see that you had more degradation of LC3, 2. So it was actually the autophagic flux increased. Also, we measured it in another way, looking at the degradation of autophagic substrate, P62. It was also more degraded, and we show it not only in cells, we also show it in liver, in animals. You can see here the reduction in LC3 and P62 in the liver, and nothing in the muscle. And I remind you that the GHF201 did not get into the muscle, so this also makes sense. It didn't affect autophagy in the muscle. So actually, what the mechanism of action that we have is actually that GHF201 is actually an inhibitor, or it reduces the level of LAMP1. Okay, so if this is the case, when we just knock down LAMP1, we should get the same effect. And indeed, if you look at the autophagic flux that I just explained, you see that knockdown of LAMP1, this is quantification of what you see here, knockdown of LAMP1 indeed increased the autophagic flux. And when on top of the knockdown, you also add GHF201, there is even a further increase in autophagic flux. Okay, so this means that the target of GHF201 is LAMP1, and the mode of action is actually inhibition of LAMP1. So when you knock down LAMP1, you get an equivalent effect. So not only was autophagy affected this way, also lysosomal function. Okay, I will get to it in a minute. Just before that, I wanted to go back, to the what I talked before, the glycolytic versus oxidative ATP production, and you can see that when we knock down, so we said that the GHF201 actually increased the contribution of glycolysis to ATP. Remember. So what we show here is that LAMP1 knockdown did the same. Okay, as you see here, okay, when you knock down glycogen, there is more relative contribution of glycolysis to ATP production. And again, when you add GHF201 on top of LAMP1 knockdown, you even enhance this effect. And you can see this effect even more when the cells are starved, as here. Why? Because starvation induces autophagy. And what GHF201 does is inducing autophagic flux, so inducing autophagy as well. Okay, and this happens even more in acute condition. Okay, because again, as we said before, we interpret this as possibly a shortened diversion of glucose from the degraded glycogen into glycolysis. The first thing that glucose does after it's actually glucose 1-phosphate is the direct degradation product of glycogen. So the first thing that happens to it is that it turns into ATP by glycolysis. Okay. So we show this nice effect on LAMP1 and on autophagy. And we actually hypothesize that this is related to the lysosomal function. So if this is related to the lysosomal function, if it increases lysosomal function, this should mean that the lysosomes should be more acidic, because in general, lysosomal are more functional when they're more acidic. So we just look directly at the lysosomal pH, and we saw the same effect as we saw on autophagy. So when you treat cells with GHF201, okay, there is an increase in the decrease in the pH, increase in the acidification. So the ratio of yellow to blue here, the higher the ratio of yellow to blue, this is what we call ratiometric staining. So the higher the ratio of yellow to blue, the more acidic the organelle. Okay, you can see that this is lysosomal associated with these confocal pictures. And then, so GHF201 has this effect. When you knock down LAMP1, you get a huge increase in the acidification, which can be even further increased by adding GHF201 on top of LAMP1 knockdown. Okay, so LAMP1 knockdown and its inhibition by GHF201 both act in the same direction. They both increase lysosomal function, and in line with that, also decrease the pH. Okay. Also, we show again, this is correlated. We saw a decrease in lysosomal size, okay, which makes sense because there's less distension, less swelling of the lysosome, because they degrade their substrate more effectively, so they don't swell as much, and therefore the size decreases. And as we saw before, also the increase in the mitochondrial membrane potential. So the waste, the waste product of the lysosomes, which is glucose degraded from glycogen, can be diverted into energy through the mitochondria. Okay. And finally, we looked at the proteomics. Okay, what happens to the proteins which are modified in the cells as a result? And this is a very thorough analysis of a few thousand proteins composing the proteomes. And you can see in this Venn diagram proteins which are either down-modulated by the disease, by APBD compared to control, again, in two different conditions, glycogen burden in red and without glycogen burden in blue. And so these proteins are down-modulated by the disease and up-modulated by GHF201. And here inversely, you have proteins which are up-modulated by the disease, and inversely, which is just correction, inversely down-modulated by the compound, by GHF201. And interestingly enough, these proteins, if we just talk about the ontology of the GO terms, if we look at functional families, okay, they actually belong to endocytosis and lysosomal function. on the one hand and oxphos on the other hand. Okay, so these are exactly the pathways, cellular pathways that we predict should be affected. So also at the protein level, the proteins which are modified by the compound are related to lysosomal function and to oxidative phosphorylation. This is very interesting because it also matched with our other results. So to summarize, what we show is actually that when you add compound A to cells, to animals, to patients, the effect is increased autophagic flux. Okay, you can see there's more fusion here. Okay, and here you can see that the binding of GHF201 to LAMP1 is shown in red, and this binding, again, in the affected the organism is compared to affected and treated. This binding reduces the lysosomal glycogen, okay, affects, improves mouse health. You can see that it's more kyphotic and much less kyphotic and more agile after the treatment. Of course, reduces polyglucosans in the tissues and increases energy level. So now just one last comment. Now, if we just move a little bit, zoom out from science, I can tell you that based on this research, a new Israeli startup company, GHF201, was established. And this is, GHF stands for golden heart flower. And this is why now the new compound is called GHF201. It is licensed to GHF. And now the GHF is conducting due diligence to invest expanding GHF201, the compounds that we produce to other storage disorders, not only APBD. And there's now a compassionate treatment in Hadassah involving 3 patients. And what I said in the beginning is that we don't, of course, we cannot inject GHF201 as a DMSO solution. Instead, in collaboration with a company called LDS, Lyotropic Delivery System, they designed a special nanoformulation, which is oral, in which the bioactive is actually at the surface, okay, of this nanoformulation. It's actually a nanodroplet or nanoemulsion. And therefore, the delivery, first of all, you don't have to inject the DMSO, of course, can be toxic and not allowed to inject any DMSO solution to humans. So we actually, the advantage there is that actually the bioactive, the nanoformulated bioactive is released from the jejunum. Okay, you can see here the binding. You can see that only the bioactive crosses the membrane. Okay, and then the vehicle, the excipient, continues on in the digestive tract. And actually, importantly, it only interacts with the jejunum, so it's very distal in the GI tract, and therefore you can bypass the portal circulation and possible metabolization of the drug or of GHF1 in this, GHF201 in this. So to summarize, I've shown you the novelty of our approach is, first of all, and I didn't stress it enough, I think, is that we have a proof of concept in patient-derived cells. So we did all this basic screen in patient cells, which reduces the translational distance. It is the first time offer a universal solution for storage disorders by inducing autolysosomal degradation. So it's a non-toxic induction of autolysosomal degradation, and since many storage diseases are related to, it can be corrected by autophagy, okay, or by reducing the burden of the whatever substance is accumulating. But you can do it by activating autophagy. So this can be a general, universal solution, at least in theory, and we also have some results for many different storage disorders. So this is also the first ever all animal level metabolic characterization of glycogen storage disease. These are the in vivo metabolism results that I showed you. So it was never demonstrated just to characterize the model, the glycogen storage disease mouse model. And we actually discover the target and the biological mode of action of our compound. So last but not least, I just wanted to acknowledge all the contributors. So we had many experiments were done in the, especially the behavioral experiments were done in the Adasa Brain Labs, headed by Professor Hannah Rosenman. And this, the cellular work and all the development, the initial development was done with collaboration with my friend Miguel Weil in Tel Aviv University. Metabolic cages were done in collaboration with Yossi Tam at Hebrew University, and the medicinal chemistry and computational chemistry were done by Benny Dadush and Tali Angel in the Robotics Center of Tel Aviv University. Mice, again, were produced by Dr. Ackman from Columbia University, and patient material was provided by Alex Lossos and Anat Mordechai. And again, we would like to thank LBS and GHF that mentioned that was to which the GHF 2.1 was out licensed. And thank you very much for listening. So if it was too long, thank you.